when I was here working at ASU, you were, I think, finishing up the superorganism, finishing up or writing the rest of the Leaf Cutters, that book. And as you were doing this, you made a point of announcing that after these book things, I'm going back to the lab, and I'm going to actually work with the ants again, and I'm going to do projects, and that's what you're doing now, like doing trail pheromone stuff with Messer and with temnothorax. And so I wonder why you keep on going back to that, and what sort of the important part of that is to you.
Well, this is the real fun. The experimental work I love to do, and it is for me not really work. It is fun, although it is sometimes tedious, and you need a lot of patience, as you know. But playing it like a detective, to find out how these wonderful creatures live, is just great fun. And I always wanted to do this. This was really why I chose this profession. But unfortunately, and this is especially true in Germany, once you climb up the ranks, you get so many obligations and you are so loaded with administrative work that when I was a young professor in Frankfurt, and I just got my 10-year professorship, I was very ready to move to the United States when I got attractive offers. Because in the United States, although once you had a major professorship, you still had enough time to do your own research. And this is what I wanted to do. Mm-hmm. So I always was interested in Messer. Mm-hmm. And the area where I worked in Arizona, down in the Portal area, Messer doesn't occur.
Right.
But as you know, I worked intensively on pogonomyrmex, trying to understand the pheromonal system and their territoriality. But I always felt Messer is a very particular case because they have this incredible column foraging strategy. Just imagine 40,000 individuals move out on one column in a day in one particular direction before they disperse, collect seeds, and then orient back to the column and home. It was reported in the literature that most likely, or it was said, no evidence exists that there is chemical trail laying on this species. And models were created to model this column foraging and the changing of directions. The lucky coincidence was that Nicola Plaus, who got her degree with Eldridge Adams, came as a postdoc working with me, and she loves field work. So Nicola and I then set out to really understand Mesopergandii. Mm-hmm. Well, it turned out chemistry plays a great role. The orientation system is totally different as we expected it. Mm-hmm. Our hypotheses were day by day thrown overboard. And finally, we got the story. And this, finally understanding the system, there is for me almost no greater joy. Yeah. So this is an explanation why I went back to experimental work.
Yeah, that's great to hear. So I want to go back and talk about sort of your origins and how you got interested in this. I know your father was interested in myrmecology. He had a fondness for myrmecophiles, or beetles and wasps that live with ant colonies. And I know he published some of that stuff. But I wonder what was his relationship with sort of academic science? Like how was that situated in his life?
Well, my father was, as it was often the case in his generation, he became a physician and a surgeon, but he started parallel to his medical studies, zoology, biology.
Okay.
And he was a full-fledged zoologist, an excellent one, incredible, diverse. But his profession became medicine, but he always maintained his great interest in biology, and he was interested. Actually, he did a medical doctor, which is basically really equivalent. Normally not in Germany. It's just 1 year of work, a medical doctor. But he did it in anatomy, and he worked with axolotl, and he worked on the endocrine glands and did transplantations. So it was really a zoological thesis.
Yeah.
But he, of course, he had a very broad knowledge in zoology. And I became very early interested in nature, in everything what moves. I was totally bored with plants. My dad always wanted to make sure I learn all sorts of plants, and I couldn't remember the names of these plants because I had no interest. But as a probably 12-year-old boy, I knew all ant species in the area in Bavaria where we lived.
Yeah.
Not necessarily by Latin names, but by popular names. And in some cases, my father invented popular names just for me to remember that I had something. When I said, what is this species? Then he sort of made quickly a name, and they stuck. This name stuck with me, and I knew them. But I have to say, we had in our house all sorts of other animals. Being a doctor in a small town, and probably there was a veterinarian, but people very quickly learned that this doctor also knows animals. And so if they found a bird with a broken wing or something, this was brought as a doctor, and my father understood how to fix the wing, and they were nursed back to health in our home and then were released. And so I kept a fox, which my dad brought home from his house visits in villages. It was a young fox, which the mother was shot, and this tiny little fox we raised, and we had dachshunds. And my ambition was to make a cross between the dachshund and the fox. But it didn't work. Yeah. And we had weasel, and I had fish tanks, so I really grew up with animals, and but I had ant colonies, and my wonderful colony, my first colony I had was a Camponotus. The European, we call it Rossameisen. It's not like the carpenter ants. Rossameisen means horse ants, because they were the largest ants in Europe. And I had this colony for a long time. I did my observations, made notes, pretending I am a scientist who sees this all the first time. And then finally, when I graduated from high school and entered university, I started out with chemistry, and in the first term of my chemistry studies, I also was in a zoology introductory lecture by a famous neurobiologist, Hans-Joachim Outram, whom we all feared because he was a choleric professor, but he was an excellent teacher. And he gave an outstanding lecture in Introduction to Scientific Zoology. And this this lecture, we have in Germany the expression "the coin dropped."
Okay.
I suddenly understood what scientific zoology is. And so my original intention also to study biology became very firm, and it became my main studies, but I continued chemistry and majored in both fields, but then continued on for my graduate work in biology.
Right.
But I should say, having been raised in field biology, and I understood as a boy a lot of forests. My father was also a hunter. However, I have to say there is a difference between the German hunters. You make lots of exams before you are allowed to be a hunter. So,
Oh, really?
Yeah.
Yeah.
You are, if you get a hunter license, you are a caretaker of the area which you use for hunting. And there are limitations what you can hunt and when, and you are supposed to take care to keep the balance. And so I was exposed to these things too. And so my first intention was, as young people, a young man wants to do what he thinks is relevant to society. And so I started out in forest zoology.
Right.
And my first papers are in forest zoology. But working in forest zoology, you are encountering ants all the time. They are used in Europe for pest control.
Uh-huh.
And then I very quickly saw, if you really want to understand what influence these ants have, carpenter ants or mound-building wood ants, you have to understand their life cycles, their behavior. And my real love to behavior came back. And then my thesis really changed to a purely behavioral thesis. And I started to take contact with Martin Lindau.
Right. Because your PhD thesis was on the social behavior of male carpenter ants.
That is correct.
Right.
And this was at that time, you always have a piece of luck also with choosing your thesis.
Yeah.
And what happened was that I saw, reading the literature, I saw in a paper by Eitman. He was a forest zoologist working on ants, and he observed, I think he dug out Camponotus nests, and he saw alates in the wintertime inside the nest, males and females. And this triggered my interest, and I said, this is the first time I have heard this. Normally alates, especially shortly after their eclosion from the pupa, a couple of weeks later they take off for the nuptial flight. The males die after mating, and the females return, break their wings, and then found colonies in different ways, different modes, depending on species. But here they overwintered in the nest, and I knew from Camponotus when they eclose. And so I picked this up and then first studied the life cycle and found that these alates, including the males, live almost a full year as adults inside the nest. And of course, this zooming in more and more what they do inside the nest, I did observe that the males, at least in the first half of their life in the nest, behave much more social that we are, what they are supposed to do.
Yeah.
And this started it, and I had the abilities to use radioactive tracers, and I learned that the males are involved in the food flow, and that they go through phases, a social phase, then a winter phase, a resting phase, and finally the sexual phase where they finish their spermatogenesis, and then take off for nuptial flights. So this became an interesting piece of work, because it led to all sorts of questions also when they, is it temperature dependent? Can they stay another year in the nest if the temperature is lower? What is going on inside the ant in the males? You know, I found they build up a huge fat body, the first phase. And then after the resting phase in the winter, they digest this fat body, and they digest literally also their gut. And I found endosymbionts in the male gut, which I a long time did not pay too much attention. Later found out that for workers and females it was already known from Blochmann, but this later was picked up again in my group, and we named them these endosymbionts Blochmannia. And I found at that time that old males get rid of these symbionts, and later, in fact, Florian Warshin, who did a diploma thesis with me and a colleague in Würzburg, discovered this in workers too. So you see, many, many years after my thesis, this was picked up. Yeah. And some of my thesis work, which I did not publish on the endosymbionts, was then published with Florian's work.
Oh, wow. That's amazing.
I still, the pictures were so good. I did at that time the histology. That we could use it. In only a couple of years ago, this paper was published in a prominent journal.
Wow.
Yeah.
That's amazing.
So, and then of course I already had contact to Lindau in between, and from a distance he helped me a great deal, and he said, "Well, finish this now. This looks all very good, and then you join my group." And I joined his group, and consider him really as my real sort of academic teacher.
Sure. Well, what you just described to me, that like all of this birthing from paying attention to a small little thing where someone reported that they dug up a Camponotus nest and found a male in winter. And like that philosophy of building on the small little things, I think is like a von Frisch Lindauer sort of thing, and that's how their whole sort of research program, the philosophy of it stems from. So, and I want to get to that in a second. When you went to, I think that was Frankfurt, right?
Frankfurt, right? Yeah.
When, I mean, science, like biology and behavioral ecology, was sort of born in Germany in 30s, 40s, and early 50s with von Frisch, Lindauer, and that whole crew. When did you, like, as a young biology student in Würzburg, either as an undergrad or working on your PhD, were they prominent figures in German biology? When did you hear about them? Like, how did their reach go?
In fact, von Frisch, of course, we knew because we learned about him already in school. And, he was in school books, and with his great discoveries he did in bees and in fish. Many people don't know that he did. Fish always worked in the summertime with the bees, and in the winter with fish. He made enormous discovery in fish, and very significant discoveries. But I remember, and it must have been 1958, because I started my university studies in 1956. There was a social insect congress, a European UC meeting in Bern, organized by the late Martin Lüscher. And I heard there was this dark-tanned, very good-looking young scientist. He just came from Brazil, and he was Martin Lindauer, a young docent, what it called. In Munich, presented his work on the Meliponian bees, on communication. This was the first time I saw Martin Lindauer and heard him. And I said to myself, this is basically what I want to do. But the situation in 1958, doctors only started to earn good money in the middle 60s. Uh-huh. Because it was still only a little bit more than 10 years after the war, and the insurance companies had no money. Mm. So although my father has a huge practice, you got only perhaps 25% of what you should have gotten from the insurance companies. And so there was no way my parents could have paid me to move to Munich and live there. And so I stayed in Würzburg, where I did my studies, which was a very, very good university. As I said, Outram was there. He, however, later moved to Munich. He became the successor of Karl von Frisch. Mm-hmm. But the first 2 years I did with Outram. And then Lindauer got a professorship in Frankfurt, I think in 1963. Mm-hmm. And I finished my PhD in 1965 and then joined this group. And I already knew some of these young people like Hubert Markel, Ulrich Maschwitz, who were with Lindauer. And it was just an amazing group in Frankfurt.
The list is
all
crazy.
The most important people now in the field really came out of this school.
Rudiger Werner, he was
Rudiger Werner was a graduate student, Randolph Menzel, Linsenmeyer, all these, very important tropical and behavioral ecologists. And it was just an unbelievable, thriving, intellectually demanding, challenging, wonderful group. And friendships we formed at that time still hold. Yeah. Some, unfortunately, passed away. One especially, Rothmeier. But I had a friendship with Markel up to now, and with Maschwitz, and so later we published papers together. We still met, and
you've published a lot of papers with Maschwitz.
Yeah, with Maschwitz, and also with Markel. I did a couple. Yes, with Maschwitz, this was always so. If we had this policy, if I see something or work on something, and independently Uli saw it too, but with another thing, we very quickly decided, let's join forces. We get more out of it if we do it together. And it was wonderfully productive and wonderful fun. Yeah. This academic collaboration, which usually is connected with a good friendship. Because you have to be good friends. You know it very well, Adrian. Sometimes you have failures, and so you sit together, and we did, these are wonderful memories for me.
Yeah, yeah. So when you went to go work with Lindauer, a lot of the people that went on to work with ants, when they worked with Lindauer, they worked with bees, of course.
Not exclusively. Maschwitz did his thesis with him, and Maschwitz was the first one who did this comprehensive study of alarm pheromones in ants. Okay. This broad comparative study. There is no other paper yet which can beat this paper, on a very broad comparative. At this time, the word was not used. It was published also in German, unfortunately, with a, at that time we still published in German with a English summary. Yeah. And so Maschwitz published with ants. Linsenmeyer did totally different things. He worked on the orientation of grabs, but Lindauer was very open. If you came and you had an interest to work and he could see that this is promising and that you have the ability, then he let you go. And he was always available when you had questions. And I remember Lindauer is this sort of more introvert person who sometimes we fresh young guys felt, oh, he doesn't understand this. When we gave him a manuscript, oh, we were so dead wrong.
Yeah.
When he took the manuscript home and then worked on it, and a couple of days it came back to me, it was full with pencil marks and question marks, and then sitting down with him. This is how I learned from Lindauer. You know, if I am able somehow now to write reasonably good papers and books, the style and this kind of thinking, how you should approach a paper, I learned from him. And this was when he was the star student of von Frisch.
Yeah.
You know, the sort of premise, you can express this in a way that the layperson is also interested. You can write a scientific paper. Admittedly, our field is easier to present in this way, but most of us don't do it. Mm-hmm. Mm-hmm. So I owe this to Martin Lindauer, and I owe to him the whole atmosphere around it. And I must say, when Lindauer sensed talent, he let you run free and made it possible for you to thrive. And my first visit to the United States, he just gave me a year of absence, leave of absence. And then when I was in the middle of my work, usually after a year, and I said, oh, this thing is so going. I wrote him, I said, because I didn't think he will continue, I said I would even cancel my position and stay another year because I got the money from a foundation in New York, and he said, no, under no circumstances continue, and so it was. I spent the first, as what is called a visiting scholar at Harvard, and
this is like 1969,
69 to 71, right? Okay. And it was incredibly fruitful.
Yeah. I want to ask you about that, but I don't want to skip over von Frisch. Like, what was his presence, and sort of how like you as a doing your habilitation work with him and the group of students and colleagues that you had.
You mean with Lindauer? I did.
Yeah, yes, right.
Yeah.
But von Frisch, as you said, was responsible for Lindauer.
Right.
And he was still around. He was retired.
That's right.
But I wonder, did you interact with him?
Oh, I knew him well, and there was this sort of custom when we did our PhD, and finally, the exam, and finally got our doctor. Once a year, the Lindauer students and the new PhDs met in Brunwinkel in Austria. This is the family estate where von Frisch lived.
Right.
A beautiful place.
And he worked after the war.
And he worked after the war, and later on had a wonderful museum there with all his collections. And he was an outstanding field biologist too, with an enormous knowledge. And so we had to do, which was more a fun part, a second exam with a master. Is von Frisch.
Really?
Yes. And he asked us funny questions, but they all were well thought out.
Do you remember one of your questions for your aunt?
Unfortunately, I don't.
Yeah.
But I remember one was whether he asked me or not, or whether he wanted to know. And he always had it sometimes in a little poem, even. He wanted to know an animal which smells with his tongue. You know?
Oh, okay.
But he had it so framed that you had to think a couple of times, what does he mean?
That's great. Yeah.
So, and then we had, it was a social thing coming together with a grandmaster, and I met him several times. I have correspondence with him up to very close to his death. He was this very fortunate person who walked his dog just 2 or 3 weeks before he died along the Isar in Munich, and was active, reading, writing all the time. So
that's great.
And I think he was 94, 95. Unbelievable high age for his generation.
Yeah.
Wow.
And then, okay, so you brought up your visiting scholarship at Harvard, which was at the end of your habilitation. And that, I think, is a really interesting period when I was sort of looking into your career and your path. Because that was your first trip over to the U.S. as a researcher.
Right.
And from what I've read, it seems like you started sort of your whole research regime here by going to Florida and studying harvester ants and camponotus, carpenter ants. And then you also went to Arizona. And that, I think, was probably your first trip to Arizona to study the ants.
That's correct. Right.
It seems like a really exciting time because on top of this is the first time you meet Ed Wilson.
Yeah.
You're at Ed Wilson, although, was working on the insect societies during that time. And you probably also met Ernst Mayr in that trip too. It seems like a really exciting portion of your life and of your early career.
Very, very so. It was really a crucial experience for me, which sort of set the route. What do you call it? You switch the direction where you go.
Yeah.
It's interesting. Ed was officially my host, and he was wonderful whenever I needed him. But at that time, Ed was very busy writing his book. We didn't interact too much because he was, in his retreat in his little office writing.
Yeah.
But he gave me all the opportunities. I could use everything he had in the lab. But there is also one person I have to mention which had an enormous influence on me was Carroll Williams. Carroll Williams is the great insect physiologist of this country. I always say he's the Wigglesworth of the United States, of equal importance. And he had an incredible group of people like Jim Truman, Fred Neuhaut, Lynn Ridiford, all the, nowadays very, very important scientists, and many others. And I interacted with this group. I went to their teas, and this is the one side, on the physiological side, where I had a background training, and Carroll Williams had his genuine interest in young people's work, and he had a very critical mind. And then on the other hand, I was totally ignorant in modern evolutionary theory. So I literally was sitting in seminars of Ernst Mayr, who became later a fatherly friend. Sometimes not always easy. He was very strong-minded, but I learned a great deal from him, and he's in many ways my model. In concerning scientific writings.
Okay.
Yes, he's very clear. He's not a poet, but he has this succinct, clear style of writing, which I admire. But I was sitting also in the ecological. There was a strong ecology and strongly theoretically inclined. Joel Cohen was at Harvard, and Bill Bosser, the young mathematician, strongly interested in ecology. I must say the math was, to me, was hard to comprehend, although I was a good student in math in school. But it didn't fascinate me so much. But I was sitting in and wanted to understand. But the approach of, also the empirical approach to ecology, Tom Schörner was still a graduate student. I think he was still a graduate student there. Yes, he was. But the seminars were very, 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 very stimulating. And so partly I was like a student sitting there. At the same time, I did my research and opened new fields for me. I had some interaction with Fred Rainier, a chemist Ed Wilson has collaborated with from Purdue University. And it just was flourishing.
Yeah.
The whole thing was, and the input, of course, then I visited Cornell, met Tom Eisner and met Bill Brown, and whom I have not met before, and loved Cornell, the Department of Neurobiology and Behavior. So these were incredible, rich 2 years.
Yeah. Who did Ed tell you to go to Florida to work on ants and go to Arizona?
I can tell you how this whole programming started, because Ed had in his insectary several colonies in big sand terraria filled with sand, and then there were Beidou's colonies.
Okay.
And I arrived in, I think it was late September or something like this. And immediately looked what is in the lab, and there were mainly two species, Solenopsis colonies and these Pogonomyrmex. So Pogonomyrmex, also they are bigger, and a lot of work was done with Solenopsis. I started to work with Pogonomyrmex in the lab, arranged that I can lead them into arenas, and began in the lab just doing orientation work. And then question came after question. And then it was, at that time, and some people still believe it today, the opinion was that harvesting ants don't have a recruitment system because they collect individual seeds, and so they don't need to recruit. But it was in the lab so clear to me when I had a denser set of oat flakes or seeds or whatever I offered in the arena, suddenly I got something what was clearly recruitment. Large numbers of ants moved over this bridge into the arena. And then, of course, what you learn when you have a Lindauer von Frisch background, you look very carefully and very detailed. And you look at the tiny things you normally ignore. And then I saw this ant which moves back shows a different behavior, moves back in the nest. And when you looked very closely, you saw the sting is coming out, and the sting is rubbed over the bridge. And then I said, they must have a recruitment system. Why do they put the sting out? And I then did the usual thing, dissected the clients, developed bioassays. Sure enough, they have recruit, which I later confirmed in the field. But they have a traditional recruitment system. They may then have additional things like encounter rates, but which of returning foragers. But we know this from other ant species. We know that returning foragers, even though they may recruit scouts, but if more come back, the response threshold to a chemical signal is lowered if, for example, we did studies in Pomeranian ants which bring back termites. And so bringing in a termite is is a signal. And it lowers the response threshold now to respond to a chemical signal when you offer it. So the encounter rate itself, I'm quite convinced, is not a recruitment signal. But it is prepares the ants to respond to a recruitment signal.
Yeah.
But you don't need always, you see, this is a misunderstanding. Once you have an established trunk root, and ants come back again from this trunk root, all with seeds, then ants may already move out, newcomers move out along this trunk route, and you don't need a recruitment system. This is an established trunk route, they come in. So this is a little sort of discourse of a discussion I had in the 70s with ecologists like Dan Jansen and Carroll, which when they thought it's a laboratory artifact that we see recruitment. And I wrote Dan Jansen at that time, I said, Dan, if you see a complicated behavior like putting out a sting and laying a poison gland trail and the ants respond to the poison gland trail in the laboratory, you better look in the field again. Because it's hard to believe that this just occurs because the ants are kept in the laboratory. And but if you follow the literature, you will see that this dispute is still occasionally going on.
Yeah. So after this initial period in the U.S. to 71, you're back in Frankfurt for a lectureship for a year, and then you applied for professorships, one at Cornell, one at Harvard. And it seems like that was influenced both by the success you had in the U.S. when you came over, and also the atmosphere in Germany at the time. I know you came back, and there was an influence of the student revolt, and it was sort of chaos. It seems like in the environment that you came from. So that I think that's probably what influenced you to apply for U.S. positions.
I did not apply. I never applied. So the thing, it's really true what you said. When I returned 71 to Germany, and I just, as I said, was a young, then very soon became, actually I was already then a tenured professor. This was just when the turmoil was brewing in the student unrest in Frankfurt. It all was connected with the Vietnam War, and there were street protests and sometimes very violent, and sit-ins and occupation of laboratories and so on. And so I may even have agreed with some of their issues. This was not the way how you get it across, And I was sitting in meetings. It's no exaggeration. In one time we had 40 hours in a week. We were in meetings, deciding how we do it.
Yeah.
And I said, if this is my profession, I chose the wrong one. And, just coming back full with enthusiasm from the United States, and now wanted to bring in some modern behavioral ecology approach, evolutionary thinking. And I was literally blocked doing this. And then when I think it was in the winter 71, I guess, colleagues from Cornell wrote me whether I could think to move to the United States and come to Cornell. And then they invited me and my wife at that time, Turit, to visit Cornell. And I think we visited 72 in probably January, February. I don't have it exactly in my head, but I think it was there. And this is what Americans do so wonderfully. They court you, really. They show you around. You meet actually on a private basis, invitations. They show me the labs. They showed us where we could live, where we could buy a house. And this was just such a difference. And I, for my visit to the United States from 69 to 71, I had no problems at all to transplant, although my English was horrible. And in school we learned only to read and write, and this not very well, but we never spoke.
Yeah.
So I hardly understood anything. Ed and I didn't understand Ed, but after a while, when you are thrown into the pot, you start to swim.
Sure.
Well, anyway, I gave 2 lectures in Cornell. I remember we stayed in the house of Tom Eisner's father. And it was clear to me, if they made an offer to me, we will come, we emigrate.
Mm-hmm.
Well, to make the story short, really, I was back in Germany. Cornell made the offer. The contract was ready to sign, and then I got an offer from Harvard, and of course the 2 institutions knew that they were after me. Mm-hmm. Now you might say, I was just beginning. I had my first science paper out, and how do they take such a young guy? It was an advantage that I got an offer from both schools. Harvard at that time gave only tenure to full professors. Cornell did not, but when Cornell learned Harvard is going to give me a full professorship, they moved up and offered me a full professorship.
Oh, wow.
So how did they come to Bert Hoelldobler? Later I heard, without giving any names, that there were 2 people at the final. Harvard has a complicated procedure to hire full professors. At this time, the final decision is not done by the department, it is the president, and he gets advice from an outside ad hoc committee. So 2 questions came up all the time to me. How did they come to this young guy, Hoelldobler? You said one thing. Surely I was now known at Harvard and Cornell for my visits. I gave talks. Yeah. And they seemed to have liked me. But nevertheless, I have to pass these 20 letters they ask around, the key behavioral biologists. And I learned later, I was told there were 2 persons in the final, an eminent biologist working on invertebrate behavior, and the young, promising Bert Hoelldobler. And occasionally Harvard takes risks. Mm-hmm. They took the risk to choose me. And why was I known well enough that this eminent behavioral biologist apparently listed me quite a number of times? They must. When they were asked for suggestions, this is the Harvard procedure. Yeah. Later on, I chaired committees like that, so I know how this works. Yeah. And, sort of what is called serendipity. Yeah. This is one of such cases. I'm not shy not to mention it. 1971 was an international ethological conference. And I was told it was the first time I attended, but it was the first time that they had a full morning with 3 plenary talks on invertebrate behavior. Mm. Normally it was always vertebrate behavior, the ethological conference. And so there were 2 eminent speakers. One was Ken Röder, the great founder of neuroethology from Tufts University, a real pioneer, wonderful man, and Martin Lindauer. and the young Bert Hoelldobler.
Oh, nice.
And I gave a talk, chemical communication was just at the beginning. And on all this new stuff on chemical communication, I did, including the Myrmecophiles. And now this talk could have gone bad. Then I probably would have made somehow my career, but I may not have made an impression so much, but the talk went very well. Mm-hmm. So I talked to Tinbergen, I talked to Konrad Lorenz, to Heinz, to Young Bates, and there were all, Danny Lerman from Rutgers University, the Institute for Animal Behavior in Newark. All these key figures in the field were there. And I assume, this is a guess, they may have been asked for, and it was particularly for invertebrate behavior, this position at Harvard. They may now have been asked. And so the name Bert Hoelldobler came up.
Yeah.
Well, so this was one of these, for me, lucky coincidences. But then, Adrian, I say it in all honesty, first of all, it was for me a really difficult decision not to leave Germany. This was clear. But should I go to Cornell or should I go to Harvard?
Yeah.
And at the end, the strengths in evolutionary biology, where I do not know yet so much, and I wanted to get deeper into that, the presence of Ed Wilson, whom I did not yet know so well, but the few times we interacted during my visit, we felt there's a good chemistry between us. The enormous ant collection, and all this, and last not least, my first wife, Turid, is an artist, and she felt correctly that Boston is a better turf for her as an artist. And so we decided to go to Harvard, and I never regretted it.
Yeah, sure, it was successful,
yeah.
Obviously.
And then, of course, this is what I wanted to say. The first 2 years were very tough, with the usual insecurities. Oh, the shoes not too large for me to fill, which they put in front of me. I had to get my grants first. I've never written an American grant proposal. I started a big introductory course in behavior, Bio 22. It was long, Bio 22. And I've never given a course in English. And my nightmare was that these very bright, demanding young Harvard students blame me for their bad grades because they don't understand me.
Yeah.
So, but things went well. I got high marks they made jokes about my accents, but they gave me high marks in their evaluation. I got my first grants. And slowly security came in, and I became part of Harvard relatively quickly.
Yeah, that's interesting. I was thinking about your experiences at Harvard and how successful you were at Harvard, and the really tight collaboration that you formed with Ed Wilson, not only in of course producing giant books like The Ants and eventually The Superorganism, but also just doing academic research with Ed. Like you did a lot on Ecophila together.
Yeah.
And from sort of a departmental view of it, why would a department want to have two people who are eminent in the same area of study, even though Ed is more sort of theory, sort of broad picture based, and you're very behavioral ecology, sort of figuring out individual things. And sort of that success of that model must have helped you sort of. work to establish what you did in Würzburg and at ASU, building a research center where you have a bunch of researchers that are eminent and very successful in sort of a narrow area of biology, and bringing those all together and sort of making the argument for how that would work and how that's a good thing to do. Like, it seems like that sort of maybe is built off of your collaboration, how successful it was and your time there.
Well, first, the position, Ed is not in a way the sort of experimental behavioral biologist working. So this is what Harvard wanted. They wanted to bring in, they had three positions, and one was in invertebrate behavior, the other was in vertebrate behavior, and the next was in primatology behavior. And mine was the first filled, and the other two were long not filled with a senior professor. We had junior professors at these, but not on a senior professorship. So this was the main motivation. Harvard looked for somebody working, and they did want to bring into the United States the sort of Convent Frisch Lindauer school, which was not established here.
Yeah.
Now, after Don Griffin left Harvard and went to Rockefeller, this field was vacant of an experimental behavioral biologist. You know, Ed did, when you think about his discovery of trailing but this was, and Ed would be the first who would agree. This is not his. Ed had really a vision and has intuition. He said there is something, but when it becomes very complicated, his strength is somewhere else, and we know all his strengths.
Yeah.
And so I introduced my course in animal behavior was certainly very different what Ed would have given a course. Ed taught ecology. He taught, a large science course for what we call the core course. And my course were the tween-level courses. We had these special tween-level courses, introduction to particular fields, ecology, population genetics, ethology, or animal behavior, neurobiology. So these were all the tween-level courses. And here students are supposed to learn the newest and the best of experimental research in the whole animal behavioral studies. And I was hired for that. It just happened. It was lucky. But at that time, it was not yet so clear with Ed and me. It happened that I work on ants or social insects.
Yeah.
Now, as I said, there was briefly the discussion whether I stay in the biolabs and get a professorial suit there where the neurobiologists were, or move, that Ed and I both move over to the new building, the MCC laboratories. Was somewhat less space, but luckily for me, Ed did never need so much laboratory space. So I had really access to quite a lot of laboratories, and I could keep, I think, 2 laboratories in the biolabs for quite a while for graduate students. But then I saw it, and Ed felt it too. It would be nice if we can move together in one floor. So Ed and I, we had separate groups, and we had our independent grants. We never had a grant together. We were clearly separate groups, and we pursued our own research. But again and again, we often had lunch together, and this is how it warmed up.
Yeah.
We were talking science, mostly science, but as when scientists sit together, they also gossip, and it became the whole thing. The sociobiology controversy started. And there were things where we both felt this is something we could do together. And like the Ecofrilla thing, because we were very complementary. And then we, good friendships develop slowly. A real good friendship developed, which even survived a real academic disagreement.
Right.
But I never felt our friendship was touched, and also my admiration for Ed Wilson.
Yeah.
So we we decided one day we go to Costa Rica. Ed has this incredible knowledge of the ant fauna. And I again come with my eye as an experimental biologist. What could we do with this species? What an interesting question you can ask with that. And again, we joined forces. I think we published 7 papers or so just from this one visit to Costa Rica.
Oh, wow.
Were all new discoveries. And we did it again. And we planned to do it more often, but then things came, with me moving away. But so we were extremely lucky. Normally, Harvard are islands of one-person professors. And we were a rarity at Harvard that 2 professors could join. There's another couple which became very, very famous, and this is Thorsten Wiesel and David Hummel at the medical school, both won the Nobel Prize. They did a lot, all this work, and they got jointly a Nobel Prize on pattern perception in vertebrates, visual pattern perception, the processing in the brain. David Hummel, this wonderful person, only passed away recently. But this was relatively rare at Harvard. And so this was a lucky coincidence. Yes, we both very much appreciated that. And that this friendship develops. I could sense it when I was a visitor, that this is, Ed and I, we were very different, but in many ways we think alike. But we have very different, our gifts are spread on different, if you have a scale, we have a good overlap. And I think we learn from each other. And so there was no intention at Harvard to do this. There was no intention to get another ant guy.
Yeah.
All what the intention was, get a vertebrate experimental behavioral biologist. And I was the lucky guy whom they have chosen. So, and the intention was really to bring this in. And so quite a number of my students, and one is Tom Seeley, I passed on the torch of this Conference Lindauer school. And, you saw like Lindauer had ant people working in his group. I had occasionally bee people.
Yeah.
Or even termite people working in my group. And I was very happy when Tom said, and I knew the problem very well. I knew Lindauer's work very, very well. I followed all this work. We were involved in discussions. When Tom came and made this suggestion, I said, go ahead. And Tom and I, we interacted a lot. Tom came, we talked it over, but it was his work. So so here was the first torch I passed on. And then when you think about many of the others who were in my group, picked up this sort of approach and in the United States. And then I reached the point after almost 18 years on the faculty at Harvard 17 years, and I said, I need more interdisciplinarity now. I need to bring stronger neurobiology in and genetics. And this is literally with NSF grants not possible.
Yeah.
I had 1 to 2 postdocs I had sometimes had 2 to 3 grants. In order to do this, I kept writing grants and grants and grants. This was frustrating too. And so when the first offers came from Europe, I had offers before, but I just was not interested. I didn't pay really attention. I said friendly, thank you.
Yeah.
But then first offer, really attractive offer, came from Zurich, and then there was a situation where, well, later on there were twice a situation that I could go to a Max Planck Institute, but I am a university person, and a very convinced university person, and I never regretted that I didn't follow up on this perhaps possibility.
Yeah.
Würzburg came, and this offer was extremely good. And it was a successor of Martin Lindauer.
Mm-hmm.
And it was, however, now the opportunity to completely start anew with how things have changed in the meantime, with a number of hard money positions, a good infrastructure. But I tell you, if the wonderful Dean Spence, the economist who later went to Stanford and then eventually got the Nobel Prize, would not have been so generous to say, Bert, I give you 2 years leave of absence, we keep your chair open, but let me know if you decide earlier not to return, so that we have the position. This was, I will never forget this generosity. I said I can risk it. Mm-hmm. Whether I transplant back into the German academic system. And the first year was not easy for me at all, and not easy for my wife too, because we had most of our friends in Boston. Mm-hmm. The boys were born in Boston, and but then I saw I could do things which I cannot do at Harvard, and in retrospect, I did not regret it. And then I got a wonderful prize from the German Science Foundation, and this almost $1.8 million, to my free, I couldn't buy a Mercedes for it, but I could use it for science as I wish.
Yeah.
And so I invited postdocs also from abroad, and we had an international group.
Yeah.
And I had the luck to recruit very good people, most of them. I made my mistakes too, but very good people who then succeeded, now all professors.
Yeah, that's great.
Around the world.
Yeah.
So in Korea, Japan, Cambridge, UK, Harvard, Cornell.
Yeah, that's fantastic.
So this is again, things come sometimes, and I think the art is that you see when a possibility comes, that you use it, or you don't use it. Sometimes it's also good not to use it, but you use it. And then, this is probably what went well. I used the right, at the right moment, made the right decision. Mm-hmm. At least I see it in retrospect. So
yeah, it seems like it to me. Yeah, for sure. Well, we're over an hour, Bert. So we should maybe stop here. But thanks so much for talking to me.
Well, it was my pleasure.
Yeah, thanks a lot.